Children, today we are going to see the topic human reproduction, lesson 2, the continuation of the topic. The first one is the mammary glands, then after that gametogenesis, in that the two subdivisions, the spermatogenesis and oogenesis. A very brief discussion of these topics. Let us see. Mammary glands, the first one. The mammary glands are said to be the sweat glands, modified sweat glands in our body. Actually, the sweat glands or the modified mammary glands is present both in male as well as in the female, whereas in male it is said to be a very rudimentary organ and non-functional, whereas in female this is functional and located in the thoracic region. And the external structure of the mammary glands, you can see the glandular portion or that is the glandular tissue with a dense fat material present here, and a median nipple surrounded by the pigmented area, we call it as the areola. Actually, the areolar gland on the surface of the nipple, they reduce the cracking of the skin on the nipple. That is why it is present here. And internal structure, when you see the internal structure of mammary glands, you can see the lobes, nearly 2 to 25 lobes are present, which is separated by the connective tissue of fat, and the lobules, the lobes in turn 2 to 25 lobes are here. They are divided into or made up of lobules, that is it's mainly by the acini or alveoli. So, the acini or alveoli, you call it as here, they are secreting milk. They are responsible for secreting milk, and they open into the mammary tubules. The mammary tubules you can see on the picture, they all join to form the mammary duct, which in turn unite to form the mammary ampulla. This mammary ampulla is finally connected to the lactiferous duct. So, that is said to be the reservoir. Finally, it ends in the reservoir, expand and it opens into the reservoir, what you call it as the lactiferous sinus. Finally, it opens into the minute pores of nipple surrounded by the areola. So, this is the internal structure. I have given it in an easy form for you to remind. Next one is the gametogenesis process. So, what is gametogenesis? The question is, what is gametogenesis? So, the gametogenesis, it is a process of formation of the gametes, that is the sperm and ovum, from the primordial germ cells by meiotic cell division, from the primordial germ cells which starts from the birth. So, the spermatogenesis, the first process here, the sequence of the events in the seminiferous tubules of the testis that produce the male gametes. So, that we call it as spermatogenesis. Genesis means the production of the sperms from the testes. So, here this undergoes 3 phases: multiplication phase, growth phase and maturation phase. So, in the first one you can see the picture here, the multiplication phase, growth phase and the maturation phase. What happens here? First one, the spermatogonia or the sperm mother cells, they develop from the seminiferous tubules. So, from the seminiferous tubules, that is said to be the site of production of sperms. So, there it develops from the sperm mother cell or spermatogonia, see here, which is now in diploid condition, 2n number of cells. The division which undergo here is the mitotic division. And the next one, here they are modified into the primary spermatocyte, which is also in the diploid condition, that means 2n number of chromosomes in here. So, this is the first meiotic division of which occurs in the growth phase. Then after that, this enters into the next stage, what you call it as the secondary spermatocyte. Here meiosis 2, in this we can find 2 number of the spermatocytes are present, secondary spermatocytes are present. Now, they are in the haploid stage, what do you call it as n number of chromosomes are present here. Then in turn, they undergo the second meiotic division to form the spermatids. See now, from each secondary spermatocyte, 2 number of spermatids will be present. So, at the end of meiosis 2 division, we have 4 spermatids are present, each one having haploid number of chromosomes. So, these spermatids are said to be non-functional in nature. So, these non-motile spermatids, they have to undergo the next stage, what you call it as spermiogenesis. So, spermiogenesis. Spermatogenesis is different from spermiogenesis. That means the transformation of spermatids into the mature spermatozoa. Transformation. See the structure of this spermatid. Actually, this resembles the spermatogonia cells, spermatogonia mother cells. Actually, here this is having the Golgi apparatus substance which transforms into the acrosomal vesicle. Also, the nucleus is here. And after that you have the cytoplasm, centriole, everything is getting converted into a transitional structure, what do you call it as the mature spermatozoa. Now, this is said to be the functional one. Okay. The next one, after this, after the completion of the spermatogenesis and the spermiation process is the next one. That means spermiation means release of the sperms. They have to release the sperms now into the cavity of seminiferous tubule. See here the seminiferous tubule cavity. Here, after the completion of the spermiogenesis, that means transforming it into a functional aspect, it is converted into, it is transformed, and after the transformation it is given to the seminiferous tubule. Okay. Now, the whole process, what happens to the spermatogenesis, how long will it take? The whole process will take about 64 days, and the sperm production remains constant at about 200 million sperms per day are produced. So, this we can see in the day-to-day life of a human being, a male. So, this is what is happening there. Okay. The next one, the structure of the human spermatozoan. Just a simple structure, let us see. Here, the human sperms are said to be microscopic in nature. You cannot see it with your normal naked eye, you can see only with the microscopic aided, and it is said to be a flagellated one. See here the flagellated one, and it is motile gamete, and this is enveloped by a plasma membrane. Completely it is covered by the plasma membrane, and it is differentiated into head, neck and tail region. And in the head you can see the acrosome and the nucleus present here. So, the acrosome, the tip of the what do you call it, the head region of the spermatozoa, it is provided with a cap-like structure which is provided from the Golgi body of the spermatid. Actually, it is produced from the Golgi body of the spermatid. Now, this contains an enzyme, what do you call it as hyaluronidase. It is a proteolytic enzyme. This enzyme is said to be a sperm lysine. Lysine means it is lysing, destructing the wall. Which wall? The fertilization membrane of the ovum. Further, during fertilization, this is useful. Next one is the nucleus. So, nucleus, the rounded structure, it is flat and a oval structure. So, now the neck region, after that head is over, now the neck region. In the neck region, which is present between the head and the body. So, here the middle piece, between that you see the neck region. Here two rounded structures, what you call it as centrioles are present. One is the proximal centriole, another one is the distal centriole. Proximal centriole, which is present just below the nucleus, it takes part in the first division of the zygote. So, after fertilization, division has to be done in the zygote. For the first division, first cleavage takes place in that. That is done by the proximal centriole. After that is the distal centriole, which is present below that. Now, this takes the axial filament of the sperm is formed by this distal centriole. Then after that you have the middle piece. In the middle piece you have mitochondria, that is providing energy rich particle, that is the ATP for the sperm, and the tail said to be the longest part. You can see the longest part of the tail, it is very slender and tapering. So, the whiplashing movements, it has to go fast, isn't it, to reach the fertilization membrane. For that, the whiplashing movement aids in the locomotion. Then after that, here also you have the longest part, what you call it as the centriole. Here the central axial filament or the axoneme, you can see this, the axoneme and an outer protoplasmic sheath, all these are formed by the plasma membrane. Okay. So, this is a small structure, you might be getting it in the 3 marks question very often. Okay. Just to learn about the structure of spermatozoa. After that, the hormones associated with the spermatogenesis. So, what are the hormones that are associated with the spermatogenesis? For everything to be done in the reproductive system, you need the association of the hormones. So, here the number one is the gonadotropin releasing hormone or GnRH, what you call here, that acts on the pituitary gland, that is the anterior part of the pituitary gland, stimulating it, that is stimulates on the FSH and LSH. That means the follicle stimulating hormone and luteinizing hormone stimulating it, and here they stimulates the growth, testicular growth, and enhances the production of ABP. That means antigen binding protein which is aided by the Sertoli cells directly that goes for the production of spermatogenesis. Also on the other side, the LH, that means the luteinizing hormone, they also acts on the Leydig cells, stimulates the synthesis of the testosterone, the male hormone. Now this also stimulates the process of spermatogenesis. A schematic sketch is given for the students to memorize easily. That is why I have prepared like this. Okay, just concentrate on this. So this picture is also helping you to understand the hormone which are aiding this. See here, this is a pituitary gland where the gonadotropin hormone is stimulating the FSH and LH directly on the testis. So you have the testis structure. So Sertoli cells, Leydig cells, FSH for Sertoli and LH for Leydig cells producing the testosterone directly. Here they are helping the production of the spermatogenesis, the sperms. Also the Sertoli cells, they produce androgen binding protein. So all this helps for the spermatogenesis process. A simple structure is explained here. So next one is the oogenesis. Oogenesis. First spermatogenesis is over, that is the production of sperm from the testis. Now let us go to the oogenesis, that is the development of female gamete or ovum or egg from the ovaries. So they are responsible for the production of ova, that is the ovaries. So here the fetal development, this is the complex process here, the fetal development in the certain cells in the germinal epithelium, the fetal ovary divides by mitosis. First it divides by mitosis, the first division always starts with the mitosis. So here what happens, millions of egg mother cells or oogonia are present. So here no more oogonia are added after the childbirth. Before the birth of the child, the oogonia are produced in millions. So after that what happens is that the oogonia cell starts dividing, they enter into the first stage. Here the prophase 1 of meiotic division. So they enter into the prophase 1 of meiotic division 1 to form the primary oocytes. So here you can see the primary oocytes which are present here. Now they are temporarily at this stage they are arrested. Okay. So first before the birth of the child, million number of oogonia are present here. After the birth, no more oogonia are added. After that, the first stage, they are arrested in this particular stage. That means the prophase 1 meiotic division, the primary oocytes, they get arrested at this particular stage after the development. Next, they enter into the next stage, that is the primary oocytes. Now they enter into the granulosa cells. Here what happened? The granulosa cells, first they are formed with a single layer of granulosa from the primordial to form the primordial or primary follicles. After that, it enters into the secondary stage. There many layers of the granulosa layers are formed. They form the theca. Theca means the external layer. Now again it enters into the furthermore stage, eventually it becomes a tertiary follicle filled with the space, what you call it as antrum. Now they have two layers, theca externa and theca interna, and inside you can see the membranes present here. Now the theca gets organized and they form the internal theca, and inside you can see the antrum fluid filled structure, and here you can see the secondary oocyte present inside. Now what happens? The picture you can find here, the oogenesis development. In the fetal period itself, so many oogonia are developed. After that, it enters into the childhood stage, it enters into the primary oocyte stage, and there it gets arrested. After that, in the reproductive stage, only primary follicle becomes secondary, tertiary. After that, the ovulation, everything happens only in the reproductive period. It enters into the primary oocyte, secondary oocyte, producing the first polar body. After that, the second polar body, everything is present only for the nourishment for the oocyte. After that, this gets disintegrated. Then only when the fertilization takes place, only if and unless the fertilization does not takes place, the second meiotic division is never completed in the female. The second meiotic division is never completed if the fertilization does not takes place, and so the egg disintegrates. So see how complex it is in the females, isn't it? Before the birth, they are developing oogonia. After the birth, in the childhood stage, they are arrested in the primary oocyte stage. After entering into the reproductive cycle, they are turning into primary, secondary, tertiary follicles. They produce a ova. Then they stop with the second meiotic division. If fertilization occurs, this goes for the production of a zygote. Otherwise, it remains as such, and the egg disintegrates if not fertilized. So, at the end of gametogenesis in females, so what happens here, the primary oocyte gives rise to only one haploid ovum. Only one haploid ovum is produced at the end of the gametogenesis, that is the oogenesis. You can see the structure here. This is the live picture of an ovum produced with the corona radiata and zona pellucida, and you have the cumulus cells, so many cumulus cells, the proliferative cells are present around that. So, the structure of ovum, very simple structure. So, usually they used to ask in 3 marks questions for you. So, here the structure of an ovum, you can see the human ovum is non-cladoid structure. What do you mean by non-cladoid? Cladoid and non-cladoid. In the first class itself I told you there are 2 different types, one provided with a shell and another one without shell. So, since this is developing within the mother's womb, this does not need a shell for protection. So, you call it as non-cladoid cells, okay, without shell. Aleacetal. When you say aleacetal, the amount of yolk which is present here is very small. So, no need of yolk because it is nourished by the placenta. And another one, it is microscopic in nature. It is very small, minute, can be seen only with the help of microscope. And the cytoplasm, here the cytoplasm is called as ooplasm here. And this contains a large nucleus. The nucleus is said to be very large, and you call it as germinal vesicle. The term here given is the germinal vesicle for the nucleus. Okay, now it is surrounded by 3 membranes. Ovum surrounded by 3 membranes. One is the vitelline membrane, zona pellucida, and corona radiata. 3 membranes. So, immediately after the ooplasm is the the vitelline membrane, the inner thin transparent membrane. And next one is the zona pellucida, the middle thick layer. You can find the thick layer here. And after that, the corona radiata, it is said to be the outermost layer, and it is full of forming a follicular coat layer. The coat formed of follicular cells. There is no gap between them, and the sperm cannot enter. More than one sperm cannot enter inside. So, to prevent the polyspermy, to block the polyspermy, the perivitelline space is present here. That means the vitelline membrane, the space between the vitelline membrane and the zona pellucida, the perivitelline space is present, which stops the or blocks the polyspermy. Okay, the next one. So, just for the children's sake, I have given this compare and contrast of the spermatogenesis and oogenesis. So, here children, you see the difference and the comparison. So, first one, let us have the comparison of this, that is in the spermatogenesis and oogenesis. Both start with the germ cells and germinal epithelium of the gonad. That is point number 1. Okay. The next one, they start with the mitosis. First process is the mitosis. They produce so many cells. One will divide into 2, 2 to 4, 4 to 16, 64. Like that, it keep on producing as many cells. And both involved in the cell growth before mitosis. The growth is done before mitosis, not after that. Okay. Next one, both involve meiosis. That means the reduction division to create the haploid cells. So, mainly this has to create the haploid cell to restore the normal number of chromosomal structure. That means the parental combination should be restored, isn't it, for that. Now, the differences is that this occurs in the testes. That means the spermatogenesis occurs in the testes and the oogenesis occurs in the ovaries. You know that pretty well. The next one, millions and large numbers are produced daily in the spermatogenesis, whereas here only one or a few produced per month. You know about the menstrual cycle that happens in every 28 days. On the 14th day only of the ovulation, the ova is produced. That is all, only one. So, here these are released during ejaculation and these are released during ovulation. And another one, this begins at the puberty, and here egg production begins before the birth and continues throughout the life, whereas here this stops at the menopause stage. And here 4 sperm made per meiosis, whereas here only 1 egg produced per meiosis. Polar bodies not produced, the division is said to be equal, whereas here polar bodies are produced, uneven division of cytoplasm is seen here, and cytoplasm is reduced in the sperm, whereas here enhanced cytoplasm is seen in the eggs. The sperm are said to be motile, that means functional, whereas that means it can move, they are locomotive, whereas the eggs are non-motile. So, I wanted you to get involved with the topics, certain topics like the differences between spermatogenesis and oogenesis, what is gametogenesis, and the diagrams along with that, representation of the diagrams for spermatozoan and structure of ovum will help you to fetch more marks this particular topic. Thank you.